up all the models for this, and he swam to shore and beyond and did an incredible job. And you see he'll have some really neat data to present that he should be very proud of. Before I invite him up, there's one small quirk that you need to know about Wasan, and that's that he has a habit of collapsing when he gives presentations. And so we've got a team of resuscitationists in the audience that'll get him back up and started on his presentation should that happen. So up he comes.
All right, thanks for coming along. I'll do my best to stay upright. Call that a win. So today I'll be describing my efforts to manipulate host cell signaling to characterize and control dengue fever. So every year there are an estimated 400 million cases of dengue, and 300 million of these cases are asymptomatic, with 100 million estimated to be symptomatic. And dengue is caused by 4 serotypes of dengue virus that share 60 to 70% homology at the amino acid level. Currently, tools to prevent or treat dengue are lacking. There is one licensed vaccine, but it cannot be widely rolled out due to safety concerns. And importantly for this talk, there are no prophylactic drugs that can be used to prevent dengue symptoms, and no therapeutic drugs that can be used to control symptoms of dengue once they have developed. So symptomatic dengue virus infection can present as mild dengue fever or severe dengue. Dengue fever is defined by flu-like symptoms such as headache, joint pain, and myalgia, or pain behind the eyes. For reasons that we don't understand, a small proportion of dengue fever patients go on to develop severe dengue, which is characterized by hypovolemic shock or hemorrhaging from leaky capillaries. Occasionally, organ failure and neurological symptoms occur as well. The disease course of severe dengue is characterized by an initial period of fever. At the period of defervescence, severe dengue patients develop a marked thrombocytopenia or drop in platelets and display the aforementioned symptoms of shock and hemorrhage. Importantly for this talk, symptoms of severe dengue usually occur at a point where levels of virus are steeply declining, or indeed the virus has been cleared. This lets us conclude that severe dengue is a result of an uncontrolled immune response to infection. So dengue virus is transmitted by the bite of a female Aedes mosquito. The virus initially infects professional antigen-presenting cells in the dermis, such as macrophages and dendritic cells, before it is trafficked to the lymph nodes. From the lymph nodes, the virus is able to infect more cells and spread systemically. Monocytes are the main cell type that is infected in the periphery. So dengue virus infection triggers 3 main signaling pathways. So dengue virus has a single-stranded RNA genome, and this viral genome is detected in the cytoplasm by pattern recognition receptors, Reg-I and MDA5. In addition, endosomal TLRs 3, 7, and 8 also respond to the dengue virus RNA genome. These various pattern recognition receptors signal via adapters to activate transcription factors IRF3, 7, and NF-kappa B. These transcription factors then upregulate the transcription of pro-inflammatory genes in the cell. And key products from this initial response to dengue are the type 1 interferons. So interferons alpha and beta bind to the interferon alpha receptor, or IFNA, that will be widely discussed during today's talk. And IFNA ligation leads to activation of the Janus kinases, JAK1 and TYK2, which phosphorylate the transcription factors STAT1 and STAT2. The phosphorylated STATs bind to specific sites in the promoters of hundreds of interferon-stimulated genes, or ISGs, that are transcribed to establish an antiviral state in the host. So we know how the innate immune response is triggered by dengue virus, and we know the nature of this response. We also know that the clinical symptoms of severe dengue are caused by a cytokine storm. So during severe dengue, host phagocytes, as well as T cells and mast cells, release large quantities of pro-inflammatory cytokines, as well as vasoactive mediators such as complement. These host factors cause a transient vascular leakage in the capillary system, and the resulting loss of volume and/or hemorrhage manifests as the symptoms of severe dengue. What we don't know is how the relatively regulated and self-resolving host immune response that characterizes dengue fever can become the unchecked cytokine storm that we see in cases of severe dengue. To be exact, we don't know what host signaling pathways become dysregulated, and also the mechanistic details of how these pathways become dysregulated. And this is what I've tried to understand during the course of my research, and this leads me to my hypothesis that symptoms of dengue can be mitigated via the manipulation of host cell signaling pathways. To address this hypothesis, I had three aims. So the first aim was to identify novel restriction factors that control host responses to dengue virus infection using in vitro assays. My second aim was to extend and confirm my in vitro findings in vivo models of dengue. And my third aim was to use candidate therapeutics to treat in vivo models of dengue. So I'll now go on to describe the work that comprises my first aim. So to dissect the nature of the host immune response to dengue, I decided to infect cells that had key immune signaling components knocked out. To do this, I took bone marrow from genetically targeted mice, and this bone marrow was then differentiated over the course of 6 to 7 days into bone marrow-derived macrophages, or BMDMs. So-called enhancing antibodies are widely used in dengue virus research. And these antibodies, raised against the viral envelope protein, can be used to opsonize virus so that it can be taken up easily by host cells that express the Fc gamma receptor. I attempted to use this method to facilitate the infection of my BMDMs. To quantify my levels of in vitro infection, I decided to use a FACS-based assay to detect viral NS1. So NS1 is a protein that is not present in the dengue virus virion. It is only produced once the virus has infected a host cell. Therefore, the presence of viral NS1 protein in host cells can be used as a surrogate marker for cell infection. And here's a FACS plot showing the levels of fluorescence that we see with infected cells as compared to uninfected cells. Here, I have infected wild-type and IFNA-deficient BMDMs in the presence of enhancing antibodies. So if I can remind you, the IFNA is the receptor for type 1 interferon. So IFNA-deficient BMDMs were very susceptible to dengue virus infection, and you can see that in the right panels. In contrast, even when antibody was used to opsonize virus, there was only a small degree of infection of wild-type BMDMs. From this, we can conclude that murine type 1 interferon is a major restriction factor for dengue virus infection. This is a point of difference to human dengue virus infections, where during mild dengue fever and severe dengue, and at time points when patients are viremic or after they have cleared virus, it is possible to detect high levels of type 1 interferon. To get around this murine-specific restriction factor, I tested to see whether I could block type 1 interferon signaling to increase the susceptibility of wild-type BMDMs to infection. So I decided to use a commercially available IFNA blocking antibody that I'll refer to throughout this talk as MAR1 to increase the susceptibility of my BMDMs to dengue virus infection. The first step in this process was to test the ability of this antibody to block type 1 interferon signaling in my BMDM cells. My readout for this assay was the detection of phosphorylated STAT1, which is downstream of type 1 interferon signaling. For this experiment, wild-type and IFNA-deficient cells were treated overnight with either an isotype control or different concentrations of MAR1. The following day, cells were stimulated for 20 minutes with murine interferon beta, and lysates were probed for phosphostat1. If you focus on the lysates of IFNA-deficient cells, you will see that there was no phosphorylation of STAT1 in response to interferon. And this makes sense because these cells don't have a receptor that type 1 interferon can bind to. In contrast, wild-type cells treated with isotype control showed strong STAT1 phosphorylation, and this STAT1 phosphorylation could be almost entirely ameliorated by MAR1 treatment. This showed that MAR1 was able to block interferon signaling in my model. So I'll now describe the experimental design for my BMDM experiments. So BMDMs were treated overnight with MAR1, and the next day, cells were infected with dengue virus serotype 2 in the presence of both MAR1 and enhancing antibody. Cells were then cultured for 48 hours in the presence of blocking antibody before cells and supernatant were processed for downstream analysis. So previously, I showed you that MAR1 was able to block STAT1 phosphorylation induced by a short stimulation of pure murine type 1 interferon. I also wanted to examine the ability of MAR1 to inhibit the interferon response to dengue virus infection. Again, you can see that IFNA deficient bone marrow-derived macrophages show no phosphorylation of STAT1 at day 1 and day 2 post-infection. In contrast, at day 1 post-infection, wild-type BMDMs exhibited strong STAT1 phosphorylation when treated with isotype, and this phosphorylation could be markedly reduced by MAR1 treatment. However, at day 2 post-infection, the same level of phosphorylation was observed in isotype and MAR1-treated samples. Therefore, MAR1-treated treatment markedly inhibited the type 1 interferon response to viral infection. However, this inhibition was lost at day 2 post-infection. In a parallel experiment, cells that were treated in the same fashion with isotype and MAR1 were harvested at 48 hours post-infection and stained for NS1 and analyzed by flow cytometry. IFNA deficient cells treated with both isotype and MAR1 were robustly infected. However, MAR1 treatment was able to markedly increase rates of infection as measured by NS1 staining in wild-type BMDMs. Rates of infection were approximately threefold higher when cells were treated with MAR1 than when cells were treated with an isotype control treatment. Now that I had established my model, I could now proceed to screening of my knockout BMDMs where key signaling pathways had been inactivated. The aim of this was to determine whether disruption of these key signaling pathways would change the nature of dengue virus infection. To assess the course of infection in the screen, I decided to assess the following parameters. Firstly, looking at viremia, which is the percentage of infected cells, and looking at cell death by a fixable live dead stain, zombie aqua. So these endpoints were both analyzed by flow cytometry. In addition to this, I wanted to look at the amount of virus that was produced by infected cells, and I decided to do this by plaque assay, and to look at cytokine production from infected cells, and this was performed by multiplex ELISA. So, as I've previously outlined, the NF-kappa B pathway is activated by both RIG-I-like receptors and TLRs in response to dengue virus infection. There are 5 different NF-kappa B subunits, 2 of which are P50 and P65. Activation of P50, P65 heterodimers causes the transcription of pro-survival and pro-inflammatory genes. In some conditions, the presence of the P50, P50 homodimer can mitigate pro-inflammatory NF-kappa B signaling. For example, Misgerd et al. Found that P50 knockout mice produce more pro-inflammatory cytokines in response to bacterial pneumonia than wild-type animals. I decided to examine the role of P50 in dengue virus infection using my bone marrow-derived macrophage model. So here are the levels of NS1 staining of wild-type, IFNA deficient, and P50 deficient cells at 48 hours post-infection. All cells have been treated with MAR1. Briefly, I should outline that for these experiments, wild-type and IFNA-deficient macrophages were infected as intra-assay controls whenever knockout cells were infected. I'll present the pooled control data as a reference for NS1 staining, as well as the data for my knockouts of interest. Here, we can see that there was no difference in NS1 staining between P50 knockout cells and wild-type bone marrow-derived macrophages. Again, for cell death, I am showing you baseline levels of cell death for wild-type and IFNA control cells, as well as a positive control, which is wild-type cells that have been treated with cyclohexamide and TNF. Here, you can see that there are no differences in cell death between wild-type and P50 deficient cells, although IFNA-deficient control cells, which are very infected, have significantly higher levels of cell death than wild-type cells. IL-6 family cytokines signal via a cytokine-specific alpha chain and the common GP130 protein. Similarly to interferon signaling, this leads to phosphorylation of JAKs and STATs. A consequence of this signaling is upregulation of the protein SOX3, which then acts to restrain further cytokine signaling in a classic negative feedback loop. To determine the effect on dengue infection of potentiating cytokine signaling, I infected SOX3 deficient macrophages in the presence of MAR1. I should note that SOX3 full-body mouse knockouts are embryonic lethal, so bone marrow from SOX3 lysem Cree mice was used to generate macrophages. The lysem Cree conditionally deletes targeted genes in myeloid cells, such as monocytes and macrophages, which are host cells for dengue virus. So NS1 staining showed that SOX3 deficient bone marrow-derived macrophages were not significantly more susceptible to infection than wild-type cells. In addition, SOX3 deficient BMDMs did not undergo more cell death than wild-type bone marrow-derived macrophages. Previously, I have described how RIG-I and TLR signaling can activate NF-kappa B. Pro-survival NF-kappa B signaling can also be activated by TNF. When TNF binds to the TNF receptor, adapters recruit cellular inhibitor of apoptosis proteins 1 and 2. These IAP proteins interact with other proteins to activate NF-kappa B. When CIP proteins are absent, TNF signaling can trigger apoptosis. Dengue is a cytopathic virus, so to determine the function of CIPs in dengue virus infection, I infected IAP-deficient BMDMs in the presence of MAR1. Surprisingly, I observed that CIP2 deficient macrophages had higher levels of NS1 staining, which is our surrogate marker for viral infection, than wild-type cells. This was in contrast to BMDMs that were deficient in CIP1 alone, or BMDMs that were deficient in both CIP1 and CIP2. And again, this suggests that CIP2 deficient BMDMs are more susceptible to infection than wild-type BMDMs. Interestingly, I did not observe any increases in cell death when I looked at IAP-deficient BMDMs. Following on from the increase in NS1 staining that I observed with the CIP2 deficient cells, I wanted to know if IAP-deficient cells would also produce more virus than wild-type cells. To determine the levels of virus produced by IAP-deficient bone marrow-derived macrophages, I set up plaque assays. To do this, I sterilely diluted supernatants taken from BMDM cultures at 48 hours post-infection. Surprisingly, CIP2 knockout cells did not produce more virus than wild-type cells, and double knockout cells produced less virus than wild-type cells. And I should note here that for this diagram, LOD represents limit of detection, and this will be an abbreviation used throughout my talk. I also looked at the production of pro-inflammatory cytokines by wild-type and IAP-deficient bone marrow-derived macrophages. At 48 hours post-infection, I found that although there were no differences in IL-6 and MIP2 production, TNF production from CIP2 cells, CIP2 deficient cells, was increased. So, to summarize my in vitro findings, I found that overall levels of cell death were very low. And the only increase in cell death that I saw was with my IFNA deficient BMDMs, which are very susceptible to viral infection. So CIP2 deficient bone marrow-derived macrophages expressed more NS1 protein than wild-type cells and produced more TNF during infection. However, they did not produce more virus as measured by plaque assay relative to wild-type cells. I was particularly struck by the diminished virus production from double BMDMs that were deficient in both IAPs, CIP1 and CIP2, as this could suggest a role for NF-kappa B signaling in assisting dengue virus replication. So as one of the most important signaling pathways in mammalian cells, the NF-kappa B pathway is often manipulated and exploited by viruses. For instance, the TAX protein of the human T-cell leukemia virus type 1, or HTLV1, is able to constitutively activate NF-kappa B signaling in infected T cells. It is thought that this is required to maintain the transformed phenotype of HTLV1 infected cells. In addition, monocytes chronically infected with HIV-1 show pronounced NF-kappa B induction and increased activation of the HIV-1 enhancer region. My in vitro results suggest that CIPs are used by dengue virus to transduce NF-kappa B signaling and establish a transcriptionally active environment in the cell which is favorable for viral replication. To test this hypothesis, I have planned validation experiments where I will use IAP antagonists or NF-kappa B inhibitors in vitro to restrain NF-kappa B signaling and confirm that a reduction of activity in this pathway can reduce viral production. I will now go on to describe the findings from my second aim of my research, which was to extend and confirm the role of host restriction factors that I'd identified in vitro in an in vivo model of dengue virus infection. So mice with intact type 1 interferon signaling are normally very resistant to dengue virus infection. And this relates to that hypersensitivity to murine type 1 interferon that I described in the first part of my talk. So I hypothesized that MAR1 treatment of our genetically targeted mouse strains would give us the best chance to observe any possible phenotypes related to signaling dysregulation driven by dengue virus infection. The experimental design for this work was to suppress these mice 24 hours before infection, and again at 2 hours before infection, and then at 24 and 48 and 72 hours following infection. So mice received 5 doses of MAR1 in total. The weight and temperature of infected mice was monitored daily, and viral titer was assessed by qPCR at days 2, 4, and 7 post-infection. So the first experiment that I performed with this model was to do a head-to-head comparison of viral load at day 2 post-infection of mice that had been treated with MAR1 or with isotype control. qPCR of plasma isolated at day 2 post-infection showed that there was an almost 3 log increase in viral load following MAR1 treatment. I should note that previously with plaque assays, I presented viral load as PFU or plaque-forming units. However, with qPCR, I'm presenting viral load as GE, or genomic equivalents. Given that this experiment demonstrated that MAR1 treatment allowed a robust viral infection, I next wanted to characterize the course of this infection in wild-type mice. So MAR1-treated wild-type mice did not show profound changes in weight or temperature. This was in contrast to IFNA-deficient animals, shown in red, that quickly lost weight and had to be euthanized at day 2 when they reached an ethical endpoint. So as I previously showed you, MAR1-treated wild-type mice had a high viral load at day 2 post-infection. qPCR of day 4 and day 7 bleeds showed that mice had largely cleared virus at these time points. Now you might think that this is not the greatest model. You get a few days of virus and then nothing. However, I should remind you that the viral kinetics in human infection are exactly the same as what I have just shown for my model, with a short 2 or 3 days of period of viremia followed by clearance, and it's during that sharp decline in viremia, or even in the period where virus has been cleared, that you see the symptoms of severe dengue. Therefore, my MAR1 model is a powerful tool that I then use to look at the effect of specific signaling deficiencies in the context of dengue virus infection. Specifically, I was interested in following up the in vitro phenotypes I had seen in IAP-deficient macrophages and translating these to an in vivo model. So MAR1 was used to treat animals that were deficient in CIP2 alone, or CIP1 lysine-cree CIP2 animals that are full-body CIP2 knockouts and have a deficiency of CIP1 in the myeloid compartment. I shall refer to these CIP1 lysine CIP2 animals as double deficient for the remainder of this talk. Double deficient animals and CIP2 deficient animals showed transient periods of weight loss relative to wild-type controls at day 1 and day 4 post-infection. In addition, double deficient animals showed transient increases in temperature at day 1 and 3 post-infection relative to wild-type controls. When I examined the viral load of these MAR1-treated mice at day 2 post-infection, it was extremely exciting to see that the double deficient animals had a decreased viral load, just as we observed with the double deficient macrophages in vitro by plaque assay. No difference was observed in the viral load between wild-type and CIP2 deficient mice. And this is again in agreement with the in vitro plaque assay results, where supernatants from wild-type and CIP2 deficient macrophages had no difference in viral load. So to summarize, MAR1 treatment of mice successfully mitigated the antiviral effect of murine type 1 interferon. Confirming our in vitro results, a decrease in viral production was observed with animals that were doubly deficient in CIPs, but not with animals that were deficient in CIP2 alone. P50 and SOX3 lysine Cree mice were also screened and no phenotype was observed. And again, this was consistent with what we found in vitro. I should note that I have planned to investigate cytokine levels in wild-type and CIP-deficient animals. We're just waiting currently for our kit to arrive, and then we'll be able to run that experiment. So our in vivo data is in line with our in vitro data, and again it suggests that removal of CIPs, and therefore reduction of NF-kappa B signaling, is able to reduce levels of dengue virus replication, perhaps due to a lower level of transcriptional activity in the host cell. So I'll now proceed to describe how I attempted to use candidate therapeutics to treat in vivo models of dengue. So this slide again. So in vitro and in vivo, a double deficiency in CIPs was able to reduce viral production in vitro and in vivo. This raises the exciting prospect that IAP antagonists could be used as a prophylactic drugs for people traveling to dengue endemic regions. Perhaps you could take a pill a day and be protected from viral replication. To test this concept, I suppressed wild-type mice with MAR1 and treated these mice with either vehicle or the IAP antagonist LCL161 at 24 hours post-infection. Mice were euthanized at 48 hours post-infection, and plasma was processed for qPCR. In addition to using qPCR to assess viral load, I was also interested in determining whether LCL161 treatment was altering the composition of the myeloid compartment in LCL161 treated animals, given that dengue virus predominantly infects myeloid cells. To answer this question, white blood cells were also isolated from blood and spleen and processed for flow cytometry. At day 2 post-infection, there was no difference in the viral load of mice that had been treated with a single dose of LCL161 or vehicle-treated animals. In addition, a single dose of LCL161 did not alter the composition of myeloid compartments, such as the monocyte cells, the inflammatory macrophages, the granulocytes, or the dendritic cells. And this data shown here is from the blood, and you can see there's no difference. And this data here is showing the composition of these compartments in the spleen, and again you can see that there's no difference. So I have shown that a single dose of LCL161 given at 24 hours post-infection was insufficient to reduce viral load or to change the composition of myeloid compartments. I am currently planning follow-up experiments that involve giving mice LCL161 earlier during the course of infection and with repeat dosing. This will enable me to further characterize the prophylactic potential of IAP antagonists for preventing severe dengue. In addition to the identification and characterization of prophylactic drugs, such as IAP antagonists, I also wanted to identify drugs that could therapeutically treat symptoms of established severe dengue. To this end, I used IFNA-deficient mice to develop an in vivo model of severe dengue. Following dengue virus infection, IFNA-deficient mice rapidly lost weight and showed a very slight increase in temperature. qPCR of plasma isolated at day 4 post-infection showed high viral loads in these animals. Multiplex ELISA also revealed that these animals produce high levels of pro-inflammatory cytokines. This was important because we wanted to see if we could use clinical stage drugs to ameliorate symptoms of severe dengue driven by a cytokine storm. To recap, many cytokines in the IL-6 family and in many other families of cytokines signal via the JAK-STAT signaling pathway. I hypothesized that clinical stage inhibitors of JAK signaling could be used to reduce excessive cytokine signaling in IFNA-deficient animals and therefore ameliorate symptoms of severe dengue. The JAK inhibitors I decided to investigate were ruxolitinib and tofacitinib. These are first-generation JAK inhibitors that broadly inhibit a range of JAKs and therefore can dampen signaling triggered by many different cytokines. Ruxolitinib is approved by the FDA for the treatment of myelofibrosis, a group of rare bone marrow cancers. Tofacitinib is approved by the FDA for the treatment of rheumatoid arthritis. Before assessing the therapeutic effect these drugs could have on my IFNA model of severe dengue, I first had to titrate these drugs to determine the optimal dose required to achieve inhibition of cytokine signaling in vivo. Infected IFNA deficient animals were given 2 doses of either vehicle or low or high doses of ruxolitinib or tofacitinib. These drugs were administered at 36 and 48 hours post-infection, and mice were euthanized shortly after the second dose of drug. You might recall that STAT3 is phosphorylated in response to IL-6 family cytokine signaling. I used levels of STAT3 phosphorylation as a surrogate marker to determine the activity of these drugs in vivo. Whole spleens of vehicle and drug-treated animals were homogenized, and Western blots were performed for phosphostat3. If you look at the lane on the far left, you can see that this is the level of phosphostat3 observed in a naive animal. The second lane shows the levels of phosphorylation observed in a mouse infected with dengue virus and treated with vehicle. Now, I will draw your attention to the lanes of infected animals treated with high doses of JAK inhibitor. These high doses were able to profoundly reduce STAT3 phosphorylation, and therefore they were the doses I selected to try to alter clinical outcomes in my model. To assess whether JAK inhibitors could improve clinical outcomes in my model, animals were infected and treated twice daily with high doses of JAK inhibitor. Mice were euthanized when they reached a predetermined ethical endpoint. From this summary data, you can see that regardless of treatment, IFNA-deficient mice rapidly lost weight and showed a minor decline in temperature over the course of the experiment. So treatments with high doses of tofacitinib or ruxolitinib were not able to reduce the time to euthanasia as compared to vehicle-treated mice. In future, I have designed experiments to assess the ability of these JAK inhibitor drugs to reduce levels of vascular leakage in these animals, and to assess the impact of these JAK inhibitor drugs on levels of pro-inflammatory cytokines. So to summarize my findings, combined deficiency of both CIP1 and CIP2 reduced viral production in vitro and in vivo. Deficiency of CIP2 alone increased NS1 and TNF levels in vitro. However, there was no difference in viral load observed when we infected MAR1-treated CIP2 knockout mice. And IAP inhibitors are currently being investigated as possible prophylactic treatments for dengue fever. So that brings me to my acknowledgements. I'd first like to acknowledge Mark for giving me this opportunity. It's been a tough PhD. I spent a year and a half trying to grow dengue virus, and I spent the next 6 months after that trying to infect cells. And there was definitely points where I was wondering whether I should do this. And talked to Mark, and he managed to, jack me up and get me through. And he's got such a sharp mind. It's really cool to be able to get a result and go and talk to him and say, look, I've found this, I think maybe this is what we should do next. And he'll say, oh, well, that's irrelevant because it doesn't fit in with the main aim, or it doesn't fit in with what we're trying to achieve with these inhibitors. And that's really cool as a student to have that sort of ability to step back and have that viewpoint. I'd also like to thank my second supervisor, Greg. Greg helped me to grow dengue virus, without which none of this would have been possible. He's been supportive. He's helped me to characterize the IFNA model of dengue virus infection, and he's also helped me preparing this talk. So a lot of thanks goes to him. Simon's also been valuable, and I enjoy our conversations with me and Simon because he's able to say, why are you doing that? And I'll have to be like, oh, well, okay, this is why I'm doing this. And he also helped me to set up the NS1 staining, which I've used for all of my in vitro work. Cody helped to run some pilot experiments of MAR1 treatment of mice in vivo. Marcel has again, been a sounding board. I've been able to bounce ideas off him. And Michael Stutz helped me to grow BMDMs, which I've used for all of my in vitro work, and Liana's helped me with mouse work. The lab's been great. They've always been supportive. There's so much expertise in that lab. It's great. You just go to anybody and you say, I've got this problem, and they say, oh, this is how you solve it. Wai Hong and Joe, my committee members, lots of input, lots of support. Stella's helped with all of my administrative work and sorting out meetings and taking minutes, which has allowed me to focus on the science. Working with dengue, a lot of the work here has been done with mice, and animal technicians have to be specially tested to work with dengue. So I'd like to thank Merle, Carolina, Shannon, and Crystal. You guys have done a lot of work, and it's been really helpful. And you're also just great people to work with. From PHI, Jessica and Esther have helped me with my bioplex, multiplex, Eliza assays. Najwa and Lorraine O'Reilly both helped me with supplying reagents, as did almost the entire inflammation division. And Cynthia helped me with some TBK1 inhibitor work that I haven't presented here today. Isabella Kong from Level 3 helped me with quantification of RNA that I've used as a standard for my qPCR. The FACS lab for all their help with my FACS work, and researchers from across Australia, from the PDI, from Flinders University in Adelaide, and from the University of Queensland for holding my hand when I was trying to establish dengue work here and providing us with virus and reagents. And Holly Romanov, my psychologist, who has helped me with sorting my brain out, I guess, and coping with everything when stuff doesn't go right. And my family and friends who are so important and have helped me with everything. Thanks a lot.
Davo.
What about other immunosuppressants like steroids or TNF blockers?
So TNF has a mixed role in the literature of dengue. Some people find a positive association between severe dengue and TNF, and other people find no significant correlation. I guess what I'd say about that is that what we were really trying to go for is a way to dampen cytokine signaling across a wide range of pro-inflammatory cytokines rather than targeting any one thing. And I know that, so I would steer away from TNF suppression for that reason, because it's not just one cytokine that's important for the pathogenesis of dengue, it's a cytokine storm. In addition, steroid treatment, from my understanding, takes a while for it to work. And dengue is quite an acute disease. The symptoms occur suddenly, and I just don't know if we can therefore use steroid drugs as an appropriate treatment for dengue because of that.
Doug and then Wai Hong.
Nice talk. There was a couple of points where I didn't quite understand the logic of why you selected certain experiments. One was why you looked into SOX3 deficient macrophages rather than SOX1 deficient macrophages when SOX1 is the primary regulator of interferons. So I wasn't quite certain about that. And then
we tried to find SOX1 mice, a colony of SOX, and we couldn't. So yeah, that was, I came in, I read a lot, and then I talked to Mark and he said, yeah, that would be nice to have, but we don't have them.
And then why you looked, the same sort of question, why you looked at phosphostat3 as an endpoint or as one of the readouts in your in vivo experiment when phosphostat1 would be the primary stat used by interferons?
So I had to think, first I actually looked for stat1. Then Mark said, why are you doing that? These are IFNA deficient animals, so you're not going to get any stat1 phosphorylation because that pathway has been eliminated.
So they were in the, I thought you were trying to dampen.
We were, sorry that I didn't make that clear. So with that model of severe dengue,
you're in the interferon.
we're in the interferon because we can't establish a severe clinical phenotype in IFNA deficient animals. So we tried to look at and see if there was severe dysregulation of different signaling pathways in our, if not sufficient animals, by reducing the restrictive effects of murine interferon by blocking with MAR1. However, although we saw the change in viral load with the double deficient animals in that model, we never saw any examples of raging fever or weight loss or any of those clinical phenotypes. So for that, we had to look at the IFNA deficient animals, and those are lacking in the type 1 signaling, so we couldn't look at phospho-step 1, we looked at phospho-step 2.
It would be interesting to look at the course of dengue in a interferon, in a SOX1 deficient animal that has a more robust interferon response, perhaps.
It's something that we'd like to do, but we haven't done that yet.
Yeah, well,
So, Wasan, in like the perfect world, and you've come through with, actually identifying a therapeutic dose modulating the infection, how would you combine that with the vaccine that's currently available but somewhat problematic, or would you see them as two very separate interventions?
It's a good question. So it's hard to vaccinate against dengue. There's 4 serotypes of dengue. They've got 60 to 70% conservation at the amino acid level. But if you're pre-infected with one strain of dengue, you can make antibodies that don't neutralize infection with a heterologous serotype of dengue virus. So currently, everybody is trying to make tetravalent dengue vaccines that are able to raise antibodies against all 4 types of dengue, all 4 serotypes of dengue, and they've managed to do this. However, raising neutralizing antibodies, which is the bar that's currently being set by the WHO, has proven to be not enough, because the licensed vaccine that exists right now, CID-TDV, it's able to raise antibodies against all 4 serotypes. However, it's not able to, in some naive patients, it's able to, vaccination puts them at increased risk of severe dengue when they're infected. And that's probably because that vaccine was raised it's got the envelope and I believe the capsid genes of the different NS1, of the different viral dengue serotypes in a yellow fever backbone. So it's chimeric, and it doesn't have the epitopes that are the main targets of the CD8 T cell response, which are actually the non-structural proteins. So there's NS1, NS3, and NS5. And NS3 in particular is targeted by the CD8 response. So it looks like we have to go back to the drawing board with that and really focus on not just the development of neutralizing antibodies against all 4 different serotypes, but also the raising of an effective CD8 response against those. So there's 2 more tetravalent vaccines that are currently in the pipeline, and they're in phase 3 right now, so we should hear back about them soon, and they have the entire dengue backbone, including non-structural proteins, so maybe they'll be able to do better. In regards to using the vaccine in conjunction with a therapeutic, I'd say that if we can have IAPs as a prophylactic treatment, maybe they'd be able to use independently of the vaccine. I don't know how. It would depend on how effective the vaccine was and how effective the IAPs were. And definitely you'd still have cause for treatment of severe dengue, because the vaccines that we've got right now only reduce the risk. They don't eliminate the risk of someone coming down with severe disease. So there'd be still be scope for severe cases of dengue and a need for therapeutics that treat the symptoms as opposed to being prophylactic.
I think we might leave it there. You did great work.
Thank you.
Thank you.